All right. Hello. My name is Mikhail Shapiro. I'm a professor of chemical engineering at Caltech. And it's my pleasure to talk to you today about how we can use ultrasound to modulate neural activity. Before I dive into that, I wanted to give you a quick introduction to our lab in general. We are interested in developing technologies to image and control the function of cells deep inside the body. This is motivated, first of all, by basic biology, because whether we're talking about neurons in our brains or microbes in our guts or immune cells and tumor cells fighting each other somewhere else in our body, to really understand how that biology works, we want to study it within the context of the intact organism. And in this case, we're talking about the brain. And in addition, we're interested in developing cell-based diagnostic and therapeutic agents that can migrate into the body and can help us diagnose and treat disease. And there have been spectacular examples of that recently with engineered immune cells for CAR-T immunotherapy or engineered probiotics for the microbiome, and potentially cells that would help with neurodegeneration. And we believe that in those cases, it's going to be important to image and control the function of these very sophisticated agents that we're introducing into the body to see where are they? Are they alive? Are they functional? Are they executing the programs that we put into them? Today, imaging and controlling the function of cells inside the body is challenging because most of our best techniques for visualizing and controlling cellular function use light. And these are things like optogenetics and fluorescent proteins, which derive a lot of their power from the fact that they're genetically encodable and can therefore connect very intimately to what's happening inside of cells. Of course, the limitation of light is that it's very difficult to get it deep into tissues. Usually it gets scattered within about a millimeter. And so it's tough to scale these technologies up to a whole brain level, especially as we're talking about mammals and larger and larger organisms. At the same time, there are technologies like ultrasound and magnetic resonance that use penetrant forms of energy, by which I mean that sound waves and magnetic fields, by and large, have no problem getting in and out of our bodies. But for the most part, these technologies have not yet been connected as well to what's happening at the molecular and cellular level. And so the big picture of what we try to do in my lab is to take these technologies and these forms of energy and connect them to the cellular level through biomolecular engineering, meaning that we find, we engineer, we evolve proteins or other genetically encodable materials that can interact with sound waves or magnetic fields. And the research in our lab quite literally breaks down into sound waves and magnetic fields and engineering various kinds of molecules or devices or biophysical methods to couple these forms of energy to allow us to image and control the function of cells. And in this talk, I'm going to only focus on the bottom left quadrant of this slide and talk about that within the context of neuroscience. How can we use ultrasound to modulate the activity of the brain? Now, the reason we and others are so interested in using ultrasound for this purpose is that it's the only form of non-ionizing energy that can be focused at depth within large tissue like the brain. This actually works not just in animal models, but in human patients. And focused ultrasound can be applied non-invasively through the intact skull and have a spatial precision on the order of a couple millimeters within humans and even finer in smaller organisms. And at that focal spot, it can be used to deliver mechanical or thermal effects. And clinically today, this is used for ablation, where you deliver enough energy that you fry a local spot of tissue, and, for example, can use that to treat essential tremor by ablating the subthalamic nucleus. And this is an FDA-approved procedure in the United States. Now, of course, in our case, we don't want to fry the brain tissue. We want to modulate it. So we want to apply ultrasound and turn on or off neural circuits. And I'm going to talk about two ways to do that. One is based on direct ultrasonic neuromodulation, and the other one is based on using ultrasound to open the blood-brain barrier and enable us to locally control things with chemogenetics. So on the first topic, this is really something that was recently brought into the scientific sphere through the work of Jamie Tyler and colleagues, who showed that you can just apply low-intensity, low-frequency ultrasound to the brain in mice and later on in humans and elicit neural responses. And this was surprising, but also exciting because you're not modifying the animals in any way. In this case, there are no genetic modifications, no chemical modifications. You're just pointing the ultrasound and you're seeing responses. And a lot of these responses look like what's shown in this video, if we can activate the video at this point. And what we see here is a rodent that is being stimulated with the ultrasound. And when the ultrasound turns on, you see this motor response. And this was exciting because it suggested that ultrasound is able to directly activate motor circuits in this case. And of course, the big question at that point was, how does this work? And I think it's very important to understand that so we can optimize this technology, figure out its advantages and limitations, and try to apply it carefully in neuroscience research. And so we embarked on a study of that question in collaboration with Doris Tsao, also here at Caltech, to look at the mechanisms of ultrasonic neuromodulation, both in vivo and in vitro. For the in vivo case, we are using calcium imaging of the mouse cortex in transgenic mice while applying the ultrasound and trying to see what's going on. And in vitro, we're working with cultured neurons where we can, again, look with imaging at what the ultrasound is doing to them, and we can have much more access to perturb them in various ways, pharmacologically, genetically, et cetera, to try to get at some of these mechanisms. And I'm going to give you a summary of what we found in both of these cases. So first of all, looking at cortical activation in mice in response to ultrasound. So what the experiment we're doing is taking skull-thinned mouse preparations, doing wide-field GCamp6 calcium imaging, and applying ultrasound to the visual cortex of the animal. This is a mouse that's under light anesthesia. And what we were hoping to see was a response in the region that we're targeting with ultrasound. But to our surprise, what we found instead is that there was a lot of activation of the auditory regions, which were not directly targeted by our ultrasound beam. And you can see that in these images here, the auditory regions are outlined in the top of the slide. And then in the bottom part of the slide, we're looking at that as a function of intensity and time. And the little circle in the bottom images outlines where we're applying the ultrasound. And so what you can see there is you get, instead of direct activation at the ultrasound focus, we were getting this contralateral biased activation of the auditory cortex. And if you apply a large enough intensity and wait long enough, yes, you will also see activation of the visual cortex, but it wasn't clear that this activation was happening directly or was arising as a byproduct of auditory cortex activation. And so if you look at this in a little bit more detail, over multiple animals, you can see consistently that you get contralateral auditory cortex activation that is happening to a higher amplitude and earlier than you get visual cortex stimulation at the target. And there was a contralateral bias, meaning that when we're stimulating on the right side of the mouse brain, we were getting more activation in the left auditory cortex. So this made us think that maybe there's some kind of auditory activation. And this was a little bit surprising because ultrasound by definition is operating at frequencies that we shouldn't be able to hear, and the frequencies that we're using here should be inaudible to mice. However, perhaps there's some kind of conversion that's happening, or there's some kind of frequencies that are part of our stimulation parameters that are not just the ultrasound frequencies, but could be audible to these animals. And so to test that, we compared the cortical response to our ultrasound, shown in the middle row there, to light, shown above, and to sound, shown below. And you can see that when we apply flashes of light, this is to the eyes, we see very nice visual cortex activation. But when we apply both ultrasound and sound, we see this pattern of activation that corresponds to auditory activation. And the fact that the ultrasound response and the audible sound response played through speakers on the outside looks similar, suggests that indeed there is an auditory side effect that is at play here. And we can actually compare quantitatively the similarity in the cortical response between the ultrasound versus sound and light, and show quantitatively that the light looks dissimilar from both of the other two, whereas the ultrasound and sound look very similar to each other in terms of the effect on the cortical activation. And that also made us wonder whether some of the movement responses that are seen when you apply ultrasound to animals could be due to these auditory effects rather than direct stimulation of the motor cortex. And the idea is that in these lightly anesthetized animals, there could be a startle response to a strong sensation. And so to test for that, we measured EMG signals in these mice while applying ultrasound, as well as light, sound, and an air puff to the front of the face as a non-auditory, non-visual stimulus. And what we saw is that while light in these anesthetized animals was not eliciting a mechanical response, audible sound, ultrasound, and air puff were eliciting similar responses. And that made us think that perhaps this is a startle type of reflex. Now, to test whether the the ultrasound is producing this kind of startle response through an auditory effect, we try to deafen these animals chemically. And so here we're doing administration of kanamycin and furosemide, where that produces a partial deafening. And we hypothesize that when we do that, the responses to sound would be diminished. If the response to ultrasound is happening through auditory pathways, the response to ultrasound should also be diminished. But the response to air puffs should not be affected because that's not happening through this deafening susceptible pathway. And indeed, that turned out to be the case. So when we look at the deafened animals, they had a reduced response to both sound and ultrasound, but not to air puffs. And so that makes us believe that we have this auditory side effect that's happening in these mice. And while we were doing this study, there was a complementary study that was happening at University of Minnesota, led by Hong-Sun Guo and Hubert Lim's lab, that was published back-to-back with our paper, where they did similar experiments in guinea pigs. And in this case, they were doing electrophysiology recording from the auditory cortex while applying ultrasound to various places in the brain, and also not even in the brain, for example, on the eye of the animal. And they saw responses that they could record regardless of where they were stimulating, which also suggested that there might be an auditory side effect happening here. And to show that this was happening through auditory mechanisms, they did a very elegant experiment where they drained the cochlear fluid from the ear of these animals and showed that the responses disappeared, so that the things that they were looking at were also likely to have been these auditory effects. So, that was kind of a somewhat depressing set of findings for us, because we were hoping that, it would just be simple and direct, et cetera. But, I think in retrospect, there's good reason to think that this should have been detectable. And that's because the pulse parameters that were used in both of these studies, and in fact were used in the majority of the published work on ultrasonic neuromodulation to that date, were repetitive ultrasound stimulation, where the ultrasound is being applied at hundreds of kilohertz frequency that's inaudible, but the pulses are being repeated at a repetition frequency of, for example, a kilohertz, which is in the audible range. And so if you take a Fourier transform of that waveform, then you're going to see not just your ultrasound frequency, but also the pulse repetition frequency, which could cause these auditory effects. And even if you are creating a continuous wave pattern that should be a pure tone, the abrupt turning on and turning off of that pattern can also lead to the generation of additional frequencies besides your pure tone frequency. And so that's something that we're trying to investigate to a greater depth, as I'll talk about in a few slides. Now, at the same time, as we were doing this in vivo investigation and getting these kind of unwelcome, let's say, results, we're also doing in vitro experiments. And in this case, we're working with cortical neurons from mice that are cultured on acoustically transparent substrates and in three-dimensional hydrogel culture, where obviously there's no auditory system. So any directs that we should be seeing from ultrasound interacting with these neurons should be direct effects. And this is work led by Sungjin Yu, a postdoctoral fellow in our lab. And what we saw here, again, using optical imaging to look at this at these responses, were nice, robust responses. So we see these calcium responses both in two-dimensional and three-dimensional culture that scale as expected with intensity and pulse duration, using parameters that match the upper end of what has been used in vivo studies. And so this suggests that there's, in addition to any kind of auditory side effect, that there's also a direct effect that ultrasound can have on these neurons. And we've been studying the mechanisms by which that is happening. And what I want to show here is just a few things that it's not. So it's not pore forming. So after ultrasound stimulation, the neurons continue to be intact. So the ultrasound is not forming pores. And it's not a thermal mechanism. We can measure the temperature changes, and they are less than a tenth of a degree Celsius. It is not a synaptic phenomenon. We can block synapses and continue to see the same kind of responses. And it probably does not involve cavitation. So for this, we have what's shown here are some voltage recordings where we're seeing the depolarization. And if there were cavitation, there was a prediction that there should be a hyperpolarization happening as well. In addition, by looking at ultra-high frame rate video recordings of these neurons as they're being exposed to ultrasound, we don't really see any kind of bubble. So at least there aren't things that are visible at the micron type of level that are involved. So this really seems to be some kind of direct mechanical interaction with neurons. And we're just finishing up a story about what mechanosensitive receptors are involved in mediating that interaction. So where we are with this direct ultrasonic neuromodulation is that there seems to be an auditory side effect that is present in addition to whatever kind of direct effects are there. And some of the motor responses that we and others see may be due to this auditory side effect rather than direct neuromodulation. So that means that we really need to look inside the brain to see what the ultrasound is doing. At the same time, our in vitro results show that we have robust direct excitation of the neurons. It doesn't seem to be toxic, and it involves specific molecular mechanisms. So there is also a direct modulation that's happening. And so the next phase of our work, and for others working in this field, is to figure out what is the set of parameters that we can use acoustically to minimize the auditory side effects while maximizing the direct neuromodulation. And hopefully we can get as much of that separation as possible. And then for whatever remains, design proper sham controls that would be needed to overcome these auditory effects. And we have a continuing NIH Brain Initiative funded project that involves several people, the investigators listed on the slide, where we're continuing to work in transgenic mice, in cultured neurons. We're doing computational modeling to see how the ultrasound field gets converted into other frequencies, shear waves, et cetera, to lead to potential auditory effects and direct effects, and human psychophysical experiments to try to document what human subjects would be experiencing and how we can design good sham controls to make this really a useful technology. So that's what I want to say about the direct modulation. And now I want to talk about how we can bring this to a more kind of molecular, engineered, cell type specific level. And the idea is that, if we're trying to study the activity of specific brain regions, or we're trying to treat a disease, it's often important not only to control the location in the brain where we're trying to modulate something and the timing of when we're trying to do it, but also to address specific cell types, because at any given millimeter-sized volume that we're targeting with our ultrasound, there are going to be multiple cell types there that are involved in different processes and pathways. And the ideal technology for neuromodulation would allow us to non-invasively control location, timing, and cell types. And the technology we're working on to allow us to do that is something that we call acoustically targeted chemogenetics. And unsurprisingly, the location targeting here is being done with focused ultrasound because of the advantages that I talked about earlier. Except here, instead of direct modulation at the time that the ultrasound is being applied, the function of the ultrasound is to locally open the blood-brain barrier to allow the transiently allow the entry of nanometer-sized particles into that part of the brain. And the particles we're introducing are viral vectors that enter the brain just at that location and are excluded elsewhere by the BBB. And they can deliver a genetic payload under cell type specific promoters. And the genetic payload in this case are chemogenetic receptors that can be activated by chemogenetic ligands like the CNO ligand that's shown here. And so the idea is that we can combine these three things together to give us access to a particular brain region, specific cell types in those regions, with timing that we can control through the chemogenetic ligand administration. And I just want to go briefly through each of these components. So first of all, focused ultrasound blood-brain barrier opening is a technology that's been developed over the last 25 years by several groups that has made really amazing progress, and we're following in their footsteps. And the key idea here is that normally the BBB comprises tight junctions that prevent most proteins and nanoparticles entering from circulation into the brain. But in this case, we administer microbubbles, FDA-approved ultrasound contrast agents that go through the blood, and wherever we're applying the focused ultrasound with particular parameters that we're using, we cause these bubbles to expand and contract and push against the endothelial wall. And this kind of massaging action causes the tight junctions to open just a crack to now allow particles that are up to a few tens of nanometers in size to enter into the brain. And after a few hours, these tight junctions close back up. And this is something that has been worked on for a number of years, and actually recently was demonstrated very elegantly in humans. I'm showing some data on the bottom right of the slide here from the Nature Communications paper that's cited there at the bottom, where that is a human brain, and the little white area there, a little white rectangle, is something that where the ultrasound was used to open the blood-brain barrier at that location, and an MRI contrast agent that's normally BBB impermeable was able to enter and provide that context. So this is a technique that works all the way from small animal models to human subjects. Now, the particles that we're introducing after opening the BBB are AAV viral vectors because they're small, and you can use them to transfect neurons. People do it all the time with intracranial injections, and you can put in your gene of interest under a cell type specific promoter if one is available. And we like AAV for those considerations, and also because recently they were one of the first types of viral vectors to be approved for gene therapy by the FDA. And the payload that we're introducing are chemogenetic receptors. I think most of this audience is going to be familiar with them. Really a beautiful technology in which you modify a receptor like a GPCR to stop responding to its native neurotransmitter and instead respond to a designer drug. In our case, we're using this class of receptors called DREADs that was developed by Brian Roth and his group that respond to clozapine N oxide, CNO, as well as other new ligands that people have developed. And so the idea in putting this all together is that we do a brief focused ultrasound procedure where we apply the ultrasound to the brain region that we want to control, open the BBB, inject systemically our AAV viral vectors encoding DREADs under a cell type specific promoter, let them enter the brain at that location, wait a couple weeks for expression to take hold, and subsequently, whenever we want to control that part of the brain, we just give the chemogenetic ligand. And the beauty of this, from my perspective, is that we only have to do the ultrasound once. And it's a 15-minute procedure. And then from then on, you don't have to walk around with an ultrasound transducer on your head to achieve neuromodulation. You just have to take a systemically available small molecule. And conversely, the small molecule therapy works like a normal pharmacological treatment, except instead of that small molecule acting on all parts of the brain that may have the receptor for it and acting in a way that produces side effects, now this drug is only going to be acting on the part of the brain and only on the neurons and the cell types that we selectively targeted through our ultrasound gene delivery procedure. So how well does this work? Well, initially we did this in mice, and we wanted to control the mouse hippocampus. One of the nice things about focused ultrasound, even compared to intracranial injections, is that to cover a certain region, you don't have to do multiple discrete injections. You can just sweep the ultrasound beam and paint the pattern of gene expression that you want. And so in this case, we're doing that in the mouse hippocampus, and we can check immediately after the procedure using an MRI contrast agent where we've opened the BBB. So we can see here that we were successful in opening it at the hippocampus. And then we inject our viral vectors. In this case, we have a fluorophore on our DREAD receptor that we can tag, and then we can look a few weeks later where we got our expression. And you can see here that we have nicely transfected the important parts of the hippocampus, and we get transfection levels that are on the order of 50% of cells at the doses of virus that we're administering here. And we can also make the cell type selective, for example, targeting transfection to only excitatory neurons. And to show that this works, we performed a behavioral experiment with fear conditioning in mice, where the idea was that we used acoustically targeted chemogenetics to put in inhibitory dreads into excitatory neurons in the hippocampus, such that if we administer CNO before mice undergo fear conditioning, we would suppress memory formation and subsequent fear recall in the mice that receive the CNO, and not in the mice that either didn't receive the ATAC treatment or didn't receive the chemogenetic ligand before undergoing fear training. And indeed, that turned out to be the case. So you can see in the plot on the left here that we're able to suppress fear recall as measured by context freezing in the recall task by more than a factor of 2. And this magnitude of this effect is comparable to what people have seen previously with chemogenetic and optogenetic treatment. So in this case, we were able to implement this technology successfully in mouse model. So the advantages from our point of view is that this is a fully non-surgical procedure. You apply the ultrasound only once. You can cover larger brain regions by sweeping it around, which becomes even more important as you scale this to larger animals, as we'll talk about in a moment. This technology works through well-established mechanisms in the sense that the blood-brain barrier opening has been worked on for some time, viral vectors have been worked on for some time, chemogenetics has been worked on for some time, and so we're combining those established techniques together, and we don't have to wonder about, you know, how is this all working like we do with a direct ultrasonic neuromodulation. Of course, the downside is that we don't, you know, control things on this kind of millisecond or second time scale that you could in principle do with direct modulation, but instead we're modulating things on the time scale of the pharmacokinetics of the chemogenetic ligand. But, you know, I think for a lot of experiments and potential therapeutic approaches, that time scale is adequate, and also gives us some room to play with because people are always developing new chemogenetic receptors and ligands. And so where we are with that work is developing ATAC 2.0. This is an NIH-funded project from the Brain Initiative that we're very excited about, where we are trying to scale this technology to non-human primates, and we think that is a system in which this technique could really shine because it's otherwise difficult to deliver viral vectors into kind of the size and distribution of brain regions that we and others are interested in doing in this type of animal model. Now, to do that, we're trying to enhance all the different aspects of our system. For example, we are engineering the viral serotypes that we're using. In mice, we used AAV9, where we'd like to develop new serotypes that are even better at entering the brain at the location of ultrasonic blood-brain barrier opening and do less in terms of transfecting other parts of the body, even though we're using them under cell type specific promoters. And this would give us a better therapeutic window and allow us to use a smaller dose of these viral vectors. We're testing new chemogenetic reagents that a lot of groups have been publishing that are very exciting, and we're going to be adapting them into the system. And trying to develop inexpensive ultrasound arrays for use in non-human primates. The devices that I showed when introducing focused ultrasound that are used in humans for ablation are very expensive and probably not something that would get disseminated widely in the neuroscience community, but there are some technologies that would allow us to make that much less expensive, because our goal is to make this that's going to be something that's going to be useful for non-human primate and other animal model research as broadly as possible. And doing all of these things requires a multidisciplinary team. And so we have the list of investigators, myself, Doris Tsao, and Viviana Gradinaro at Caltech, who are working on the viral engineering and implementation, initial implementation in non-human primates. Charles Caskey, Will Grissom, and Limin Chen at Vanderbilt, who are experts in focused ultrasound and using MRI to visualize and control that ultrasound, as well as non-human primate neuroscience, which will allow us to validate our approach in 2 different labs, 2 different animal models, to make this really a robust technology. And this is something that this very multidisciplinary project is something that is specifically enabled by the Brain Initiative, and we're very grateful for that support. So, to summarize, I talked about this bottom left quadrant here, where we can use sound waves to control neural activity, and I showed a couple of different ways of doing that. The broader statement that I'd like to make for people that are interested in this field is that recently it's also becoming possible to use ultrasound to visualize neural activity. In a technology that's available already today, you can do that through hemodynamics with functional ultrasound developed by Mikhail Shapiro and colleagues in Paris. And we are also developing molecular tools that allow us to look at things like gene expression and potentially more dynamic signals using ultrasound instead of light. And some of the references for that work are listed in the top left. So with that, I'd like to thank my lab and our collaborators and our supporters, especially the Brain Initiative. This kind of work takes a big team of multidisciplinary people to pull off, and so I'm grateful for that. I'm grateful to everybody who is watching this, and I welcome questions. If you'd like to do that, feel free to get in touch with me via email. Thank you.